We're very pleased that Barry Karger joins our list of esteemed winners of this award. Barry Karger is the Director Emeritus of the Barnett Institute, which he founded in 1973, and the James L. Waters Emeritus Chair and Distinguished Professor Emeritus at Northeastern University in Boston, Massachusetts. Over the 60-year span of his career, Barry Karger made an enormous impact on the field of separation science. He began his career in gas chromatography, but with the advent of liquid chromatography in the late 1960s, he quickly became an early leader in that field. Along with Chava Horvath, Barry Karger was one of the first to develop reversed phase LC, which, as we all know, is the predominant mode of LC today. As part of that work, he developed sensitive methods to evaluate bonded phases, which led to improved method reproducibility and overall performance in reverse phase LC. Also, in collaboration with Lloyd Snyder, he introduced a means of classifying mobile phases in reverse phase LC, leading to better understanding of cell activity. Going beyond reverse phase, he also shed light on the basic principles of hydrophobic interaction chromatography, or HIC. He was also a pioneer in capillary electrophoresis. And in 1989, he started the first conference series on the technique. And that event is now called Microscale Bioseparations, or MSB, and is still held annually. But Barry Karger did not limit himself to fundamental developments. He also significantly advanced the practical application of analytical techniques. One of the most famous of these was the Human Genome Project. His research in CE led to the first demonstration of replaceable linear polymer matrices, a key advance in automating DNA sequencing. His linear polyacrylamide matrix was used to sequence more than 40% of the human genome in 2001. The other major area of application where he focused was protein separations and biopharmaceutical analysis, areas where he also left an enormous impact. For example, he was active in the development of peptide mapping by reverse phase LC to characterize proteins, certainly an essential tool at that time as the field of biotechnology was emerging, and which continues to be an essential tool today in the biopharmaceutical industry. In the new millennium, his focus shifted to coupling mass spectrometry to LC and CE for protein analysis and proteomics. He recognized the importance of low flow rates for achieving high sensitivity in electrospray ionization, MS. He also devoted a significant effort to developing narrow bore LC columns for online MS coupling. He also applied these developments in LCMS and CEMS to biopharmaceutical characterization. In one example, his group, using LCMS, was able to elucidate the disulfide linkages in a protein with a complicated cysteine knot, where a total of 15 possibilities existed. In another, they characterized beta interferon using CEMS, achieving over 400,000 theoretical plates and identifying 138 different modifications of the intact protein. In total, Barry Karger is the author of 375 publications with an H-index of 98 plus 50 patents, and he has given some 500 oral and poster presentations, including some 50 plenary lectures. But Barry Karger's influence was not restricted to his research. It also arose from his work as an educator. He trained more than 500 PhD students, postdocs, and staff scientists, and he trained so many people who became leaders in analytical chemistry. People like Wolfgang Lindner, Nobuo Tanaka, Heinz Engelhardt, Frank Foret, Howard Barth, Peter Schoonmakers, and many more. He was known for being generous in his support of his students during their studies and throughout their careers. In his lab, he developed a positive and stimulating atmosphere, and there was a lot of creativity and teamwork. As Peter Schoonmakers said, he was creating the kind of atmosphere that hip companies from the US West Coast are now known for. Given his remarkable career, it's not surprising that Barry Karger has received numerous awards. There are far too many for me to mention here, but I'll name just a few highlights. The Dal Noguera Award, the ACS Award in Chromatography, the MS Sweat Medal, the Martin Medal from the Chromatographic Society of the UK, the EAS Award for Outstanding Achievements in the Fields of Separation Science, the ACS Award in Separation Science and Technology, the Halash Medal, the Cass Award for Distinguished Contributions in Separation Science, and the Golay Medal. Ladies and gentlemen, for all of these reasons, it's my great pleasure to present to Barry Karger the 2022 LCGC Lifetime Achievement in Chromatography Award. Barry, congratulations.
Thank you. let me say I'm honored to receive this award and would like to thank LCGC for selecting me. All right, so this is the crystal that would normally be given to me at the PitCon meeting. So thank you very much. And let me also congratulate Jim Grinias for his award. And of course, the award also recognizes all the students, postdocs, and colleagues and staff who collaborate with me. And I have many wonderful memories of the interactions. One of the things I do is I try to keep in touch with many of my former students, and I'm always interested in their successes. So now let's go on to the talk. I'm honored to have received the LCGC Lifetime Achievement Award. And today, for my talk, I'd like to provide a perspective on separations and analysis in the life sciences over my career. I began at Northeastern University in 1963 as an assistant professor and continued until 2016 when I became emeritus. And since that time, I have remained active in the life science area. What I'd like to do is provide a personal perspective on analytical separations during these 60 years. And then I'd like to turn to where we are today and suggest where we are heading in the near future. Finally, I'd like to say a few words about analytical chemistry in general and provide some advice for the coming generation. Before I begin, I need to acknowledge all the PhD students, postdocs, Barnett faculty and staff, all my collaborators over the years, and colleagues. I have had over 250 PhD students, postdocs, and staff work with me, and I'm proud of each and every one of them. Let's first look at the early days, back in the 1950s and a little bit earlier than that. In 1948, R. Tiselius won the Nobel Prize for electrophoresis. And then in 1952, Martin and Singe were awarded the Nobel Prize for partition chromatography. Then in 1955, Perkin-Elmer introduced the first GC instrument, the vapor fractometer. And in 1958, Marcel Gaulet published his paper on capillary gas chromatography, showing the fundamental basis of separation with the capillary system. And with all of these events, we can say that the early 1960s was the beginning of the modern age of chromatography. So if we look from 1960 to the 2020s, over the 60 years, separation science has had an enormous impact in the life sciences, the biology, pharmaceutics, biotechnology, clinical science, and many, many other areas. Where would we be without it? Analytical and preparative scale, of course, have been used. And we can say that these years could be called the golden age of chromatography and electrophoresis. Returning to the early days, chemists and biologists were really in different worlds. Biology had not yet become a molecular science. Liquid-liquid extraction was widely used to obtain purified or semi-purified materials. And in order to improve performance, multi-stage devices were developed and utilized. I remember well the Craig machine developed by Lyman Craig from the Rockefeller Institute. Paper chromatography was widely used. It was invented by A.J.P. Martin. Liquid chromatography was really a slow process based on gravity feed. And indeed, a PhD thesis could be awarded if one isolated one or only a few proteins. As I said, gas chromatography was emerging, and there were theoretical discussions about the difference between packed and capillary columns. Quantitation involved using a polymeter where we simply drew across the peak, or we just cut and weighed the paper. There were no computers at that time, and we used a slide ruler for calculations. Many of you probably don't even know what a slide ruler is. And there was no mass spectrometry. In my PhD thesis at Cornell University with Don Cook, between 1960 and 1963, we studied the difference between packed and capillary columns. And here is a figure comparing the two from a paper we published in 1964 in Analytical Chemistry. It's also interesting to note that James Lovelock in the early 60s published a paper on the flame ionization detector, which he developed. And I built a flame ionization detector as part of my thesis. So in the 1960s, the late 1960s, interest in liquid chromatography, high-performance liquid chromatography, or high-pressure liquid chromatography, began to be developed. And we and others began looking at liquid chromatography. And here is a paper we published in 1971 in Clinical Chemistry, where we used liquid-liquid chromatography, where we coated onto the packed beads a immiscible liquid. Here we're separating steroids, and you can see the poor efficiency that one obtained in those days. What's interesting is we took standard steroids and made a mixture. And actually, we were separating male and female steroids together. And this shows how much we knew about biology in those days. So the 1970s saw major advances in liquid chromatography. There was the conversion from liquid-coated columns to bonded phases. And they were actively developed during the 1970s. We started off with particle diameters in the 20 to 30 micron and going down to the 5 to 10 micron at the end of the decade. It's interesting to note that one of the first packing materials was Zypex, developed by Jack Kirkland at DuPont. And this was a 20 to 30 micron particle with a core shell, so that the diffusion within the pore was minimal relative to diffusion throughout the whole particle. Over the decade, there was an improvement in robustness, both in terms of the packing of the columns as well as removal of silanol groups on the bonded phase. And there was a broad development of small molecule HPLC. And then at the end of the decade was the beginning of peptide separations. The fundamentals of separation by chromatography, electrophoresis, and so forth was rapidly developing. And Lloyd Snyder, Csaba Horvath, and myself felt it would be important to put a book out discussing the fundamentals of separation and describing the specific separation methods available at that time. So in 1973, we published the textbook on separation science. This book became the standard graduate text for separations for over 25 years. The first part of the book dealt with separation fundamentals, and then the individual separation methods followed. It's interesting that mass spectrometry had 2 pages. It was discussed at the end of the book under the chapter Miscellaneous Separation Methods. We did a lot of activities in the 1970s, but let me just show you one slide from that era. This was published in 1979 and shows the first separation of optical isomers by direct injection of the optical isomers into the liquid chromatographic column. Here we used a optically active metal chelate additive for separation. We didn't continue in chiral separations after that. However, one of our co-authors, Wolfgang Lindner, who was a postdoc with me at that time, did continue and of course became, over the years, a major figure in chiral separations. Turning to the 1980s, this is an era when peptide mapping was developed. And it was the beginning of the analysis by reverse phase chromatography of proteins. Size exclusion was also widely used, both for proteins and, of course, for polymer analysis. Hydrophobic interaction chromatography using a relatively high-performance system was introduced, and key to this was collection of material in an active form. And of course, HIC is very important for preparative scale in the field of biotechnology today. 5-micron particle size were routinely used. Now, LCMS became widely available, but mainly for small molecules and synthetic drugs. No one thought in terms of the potential for doing high molecular weight proteins at that time. Key to the 1980s, of course, was the start of biotechnology and the beginning of capillary electrophoresis. As I said, the biotechnology industry began in the 1980s, and obviously there became a critical need for product characterization. And since these were protein therapeutics, peptide mapping needed to be developed and in a highly robust manner. The beginnings of glycan analysis began because, for example, monoclonal antibodies were being developed at the late 1980s, which had carbohydrate groups. Preparative chromatography had major advances needed for the purification of the proteins. Now, rapid growth was anticipated for the biotechnology industry, and there weren't people available who had experience or knowledge in these areas. Where were we going to get these people? So, in the mid-1980s, Fred Renier and I formed a company called Bioseparations Incorporated to train people working at the companies in the these fields of protein characterization. And so we went to different companies like Genentech, Eli Lilly, and so forth to provide training in both peptide mapping by liquid chromatography, and also in the late 1980s talked also about capillary electrophoresis. We also introduced intensive seminar programs beginning in 1987, and we introduced the term analytical biotechnology. And interesting, some of the questions at that time: can HPLC or electrophoresis detect an amino acid substitution in a polypeptide? So as I said, peptide mapping was rapidly developed into a robust tool in the 1980s. And here's an example at the end, 1988, published by Rob Garnick and colleagues at Genentech in the peptide mapping of TPA. And you can see the high reproducibility obtained in the two chromatograms. Interesting, the arrow in the bottom chromatogram shows a substitution of glucamic acid on the peptide. So yes, we could see the substitutions. But in order to determine what each peptide was, we had to collect it, or they had to collect it, and then do Edmund sequencing. So it was an enormous job. The 1980s also saw the rapid increasing interest in high-performance capillary electrophoresis. After the lecture by Jim Jorgensen at the HPLC meeting in Avignon in 1982, we began to work in this field in the mid-1980s, and in order to advance the field, we organized the first HPCE meeting in 1989 in Boston. The meeting continued to grow and expanded and has reached the 37th meeting, which was held in 2021. The meeting is now called Microscale Separations and Bioanalysis. As many meetings, unfortunately, the one in Boston was held virtually in 2021. Now, turning to the 1990s, a major separation problem was DNA sequencing, and the Human Genome Project took a decade, the whole 1990s. CE continued its development, and the coupling of liquid chromatography to mass spectrometry became widely available. Both MALDI was utilized as well as electrospray. And the broad applicability of high pressure liquid chromatography became widely used. So over the years, we developed linear polyacrylamide, which was used, one of the polymers used for the sequencing of the human genome, which was completed in the late 1990s and published in the early 2000s. And here we show an example of 1,300 bases sequence. We generated 10 million theoretical plates for the column. It's interesting, it cost $3 billion to sequence the first human genome. And now one can do it for $500 or less, which shows the enormous advances that have been made over the 20 years. At that time, we all thought DNA was the key molecule. We thought RNA was just simply a messenger. And of course, as we'll see, RNA now is the exciting molecule. And a summary of capillary electrophoresis with the Genome Project was published in Electrophoresis by myself and Andras Gutmann. Let me just take a moment to talk about 10 million theoretical plates. So if we look at the resolution, and let's say we want a resolution of, let's say, only 0.7, we would be able to get that for molecules that differ in their electrophoretic migration of only 1.001. So efficiency is very important, but efficiency alone did not solve the problem. It had to be integrated with the detection, it had to be integrated with automation of 96 capillaries, and the ability to blow out the matrix and reload it and get the same separation. In other words, to be able to have automation. Turning now to the 2000s, LC-MS became, of course, now available for peptides and also for proteins. And the beginnings of proteomics, looking at the separation of thousands of peptides in one run, began to be developed. Metabolomics also became usable. And biotechnology now had rapid growth with the introduction of product after product. Interesting, HILIC was developed. It was actually introduced well before the 2000s, but here it became commercially available and utilized by a large number of groups. And if we look at the packing materials, the ultra-high pressure liquid chromatography was developed. So sub-2 micron particles, core shell particles, which are a little bit, let's say, bigger, and using pressures of 1500 bar and even a little higher. We were active in the development of methods for analysis of proteins by LC-MS in the early 2000s. And here's one example from work we published in 2006 in molecular cellular proteomics. Here we show the characterization of epidermal growth factor receptor, a protein of 170,000 Daltons. We did this by peptide mapping, and we were able to also determine 13 phosphorylation sites along with glycosylation. Turning next to the 2010s, and of course 2D and 3D LC-MS became widely available for deep characterization of monoclonal antibodies and other protein molecules. Microfluidics now became commercially available and utilized. And the introduction of top-down proteomics and the characterization analysis of proteoforms began in earnest. Glycan analysis, and at the end of the 2010s, the beginning of lipidomics started. CEMS was introduced, utilized, And of course, one of the big differences from earlier eras is digitization and automation of methods. But an interesting question began and still is being asked. Is chromatography a mature field? Will there be any more advances possible? I only have time to take one example from the 2010s, so we'll take a quick look at microfluidic separations, systems that were commercially introduced. The first, developed by Gert Desmet, is the micropillar array that was introduced by Pharmfluidics. Instead of using packed beds, one uses pillars that are microfluidically manufactured. And this now is, Pharmfluidics is now part of Thermo Fisher. The second system is a CEMS system, microfluidic, the zip chip, developed by Mike Ramsey and introduced by 908 Devices. And this now is utilized for analysis certainly in the biotechnology industry. And the third is a high-resolution mobile ion mobility system that was developed by Dick Smith, the SLIM system. And this now has been commercialized by Mobilion. Having scanned the previous 60 years, we now want to turn to look into the 2020s and the future. We're all well aware of the increasing importance of artificial intelligence, deep learning methods, for a variety of applications. What I want to point out is it already is having an impact and will have an increasing impact on separations and analysis. We have always been interested in developing methods to predict retention based on structure. Now, using deep learning methods, we have very powerful tools to predict retention of peptides, and more recently, methods are underway for prediction of retention for intact proteins. As we'll see, we're very interested in high sensitivity analysis. And one of the potential tools for this is the use of artificial intelligence to reduce noise in detection. There are advances in mass spectrometry. I've already mentioned ion mobility, and the resolution for ion mobility keeps improving. Also, the ability to measure high mass with high resolution is now available. And so there is a need for megadalton separations, the separation of native intact protein species. We're all aware of the great interest in RNA analysis today, both basic and therapeutic. And of course, this has been pushed very rapidly by the pandemic with the mRNA vaccines. And also single-cell methods, particularly proteomics, but more generally omics, metabolomics, and so forth, are of high interest today. High-resolution ion mobility is becoming more and more important. And several companies have instrumentation for this at the present time. Mobilion, I've mentioned, and also Waters, for example. And of course, Bruker has the TIM system, TIM's TOF system, and Thermo has the FAME system. Here's an example from MobileIron published last year in JASMUS, and you can see that the SLIM system then is coupled to a TOF. And here we can see the analysis of deamidation forms resolved by high-resolution ion mobility. One other example where high-resolution ion mobility is useful is taken from work of Jared O'Clair at the Biotechnology Training Center at Northeastern University. Here he is separating a protein by native conditions, and what one can see is that the ion mobility allows the separation of different conformers of that protein. Okay, turning next to the field of RNA therapeutics. We all know that this is an extremely hot topic, and we can anticipate major growth in the years ahead. And of course, as I said right at the beginning, the ability to characterize the product is essential, without which you can't have a product. So, and of course, today, where would we be without the RNA therapeutics in the vaccines that we have for the COVID pandemic? And so we see here that we have a vector surrounding, in this case, DNA, but also we could have RNA that then goes into the cell and, of course, releases the RNA for its ability to either block production of proteins or for production of proteins within the cells. And we have a lot of different products potentially that can be utilized. We already have antisense DNA, antisense oligos, which are being widely used commercially. We have siRNA, which are small RNAs for blocking expression. We have single guide RNA for CRISPR-Cas, and that's now being commercially developed. And of course, mRNA in the vaccines, and then potentially also for gene therapy and so forth. And then there are other types of RNAs people are looking at. So, for example, self-amplifying mRNA and circular mRNA, so that one can continue to use the mRNA for production of protein within the cell. And it's interesting also, we need to note there are over 150 RNA modifications that we're going to have to be able to analyze. And the field of epitranscriptomics, where we methylate different different nucleotides, is significant. And so RNA analysis is critical, as well as the vectors. Okay, so here's a slide that I borrowed from Jonathan Bones at NIBRD in Dublin. And this shows the different LCMS-based methods to monitor the critical quality attributes of AAV caspids. AAVs are used as vectors. So we first are interested in looking at the intact capsids, which are high molecular weight species, and we want to do that. We're able to look at that in intact. Then we can, of course, do the digestion of the proteins and look at that at the peptide level. Okay, But I want to point out that we are trying to do analysis of very high molecular weight species. As an example of the analysis, I've taken this slide from a paper published by Jonathan in Analytical Chemistry in 2021. One of the questions involved in this type of analysis is the packaging. Is RNA indeed within the capsid, or is the capsid empty? And the relative amounts of each. On the left-hand side, in the mass spectra that we show, the capsid was diffused within the high molecular weight mass spectrometer, and Jonathan was able to show that he could analyze both the empty and the filled without pre-separation online. On the right, we see the separation by anion exchange chromatography of the mixture. Here, the mobile phase used for this separation did not allow input into the mass spectrometer. However, this does show the potential. Here, Jonathan used non-porous particles, beads, for separation. Potentially, wide pore particles may be possible. Additionally, open tube potentially can be used for these megadalton type separations. Not only are we interested in the caspids as vectors, but of course we all know about lipid nanoparticle vectors being used widely, for example, with siRNA and now with mRNA in the vaccines. And of course this creates major challenges analytically as well. And so there are real opportunities with respect to separations of different lipids for the characterization of this vector. Okay. As open tube separations, we immediately think of capillary electrophoresis. And so here I've taken an example from the work of Alexander Ivanov and Anne-Lise Marie of Northeastern University. Here we show the native CEMS of GRO-EL, a tetradecameric protein complex, with GRO-ES, a heptameric complex, along with ATP gamma S. And the total molecular weight is roughly 1 megadalton. Here you can see some separation, and then the mass spectrum is shown. So there clearly will be opportunities here to think about using capillary electrophoresis for some of these applications. Turning now to RNA analysis, here's an example from last year by Kelly Zhang in the analysis of single-guide RNA, which is part, of course, of the CRISPR-Cas system. The strategy here is to use several different ribonucleases to digest the RNA. The RNA here is 100 nucleotides long. And after each digestion, there is separation, here using Hillick for separation, followed by analysis. A different approach for RNA analysis by LCMS is shown in this slide. This is taken from a very recent paper published in bioRxiv and by Dick Mint from the University of Sheffield and Cook from Thermo Fisher and colleagues. Here they take one ribonuclease, RNase T1, attached to a magnetic bead, and they control the time of digestion in the range of roughly 5 to 15 minutes. In this way, they get partial digestion and longer fragments. Since we only have 4 bases for RNA, if we go to shorter fragments, we would have difficulty where to place them in the sequence. This is analogous, this separation is analogous to what happens with peptide mapping. Here, the separation uses ion pair reverse phase chromatography. We can anticipate that there will be significant advances in the coming years for RNA analysis by separation mass spectrometry. Turning now to another area, many of you are aware that single-cell proteomics is becoming very active at the present time. And we can anticipate that it will expand rapidly in the years ahead. Here's an example that I've taken from Nikolai Slavov from Gene Biology last year. Nikolai is on the faculty at Northeastern University. And here he is using TMT, the bar-coded approach, to do multiplexing for the single-cell analysis. You can barcode individual samples and also include a reference, and then using 50 or 100 cells as a carrier to minimize adsorption losses on surfaces. These are mixed together, and then digestion and peptide separation, followed by LCMS. And using this approach, thousands of proteins, can be determined in a high throughput manner so that 1,000 cells can be analyzed in a reasonable amount of time. I want to point out that he's using 75 micron columns, and the flow rate is roughly 125 nanoliter per minute. Now here, separations using low flows become very important. And here I've taken an example from Dick Smith's group back in 2002, showing the influence of tube diameter on sensitivity in LCMS electrospray. And you can see that as you go to narrower and narrower tube diameters, or really lower and lower flow rates, you're getting higher and higher sensitivity. And we were very interested in this over the years and showed high sensitivity for limited sample amounts using 20 micron monolithic columns. And then also we developed 10 micron ID plot columns. Today, instrument companies have pumps that can go down to low nanoliter per minute, but they recommend above 100 nanoliters per minute generally, as was seen in the previous slide. So with advancements in pumping systems to be able to robustly work at lower flow rates, the opportunity for even higher signal enhancement can occur. And we can move not only from single cell, but organelle analysis as well, potentially. I'd like to now turn to talk to the younger generation. First of all, I want to look at analytical chemistry. And I've always said it has 3 M's: molecule, measurement, and meaning. We need to understand the chemical properties of the molecule. Obviously, we need to be able to handle the measurement, all the principles involved and so forth, and why we're doing the measurement. And finally, what do the numbers mean? You know, what is the purpose of the analysis? And when I talk to some of my former students today, they say, you almost have to be a Renaissance man because you have to know so much. But it's not as bad as you think because you have the internet. So you'll be able to look things up quickly. And so you have to not just pigeonhole yourself in one of the 3 M's, you have to be in all 3. And the way I see it, analytical chemistry has never been so important. The fundamentals of the 3 M's need to be taught in the universities and training centers. There is a gap in the basic knowledge of many workers. This is especially true in the separations and MS fields. So what I've learned, advice to the next generation. I've already said the 3 M's, which means you need to understand the problem and the fundamentals of the measurements. Change continues to accelerate, and you're going to be forced to reinvent yourself several times during your career. You must not be afraid of change. The issue of basic versus applied, or the term sometimes translational, you have to be able to move back and forth between the two. Of course, data handling is very important today, and artificial intelligence. You need to be comfortable in these areas, and I think all students going into these fields need to have a basic understanding of data processing and artificial intelligence. And you need to take a holistic view of the analysis and look at all of the steps, because each step may affect other steps. And as we had in the early days of biotechnology, a major issue today is shortage of people, because biotechnology is growing very rapidly at the present time. Nevertheless, the future is bright. So, in closing, let me thank the Karger Mafia. That's the way we used to always call it. I'm proud of all the accomplishments of the PhD students and postdocs. And I have many memories of the collaborations with people and the staff scientists I've worked with. And with colleagues, I've learned much from interactions. I've listed here some of the key players in chromatography over the years who are no longer with us. And I have memories of all of them and of the time spent together. It was fantastic. And I thank all for what was achieved. I feel so lucky. The next decade and beyond will lead to discoveries we can only dream of today. I wish I were younger.